Everyone present. Today I am Debopama Chowdhury and I am going to present my project titled Design and Development of an Automated Portable Bioreactor for Algae Cultivation. So coming to the reason as to why we are shifting to a portable algae bioreactor, our main aim and our final end product is to produce bio oil from from the algae which we are growing. So the traditional methods of the insulating oils are basically used for transformer cooling purposes. So the traditional ones were the mineral oils, first generation, second generation oils. The problem with all these three are the mineral oils were made from fossil fuels. Of course, we cannot use fossil fuels because they are depleting. The first generation oils were made from edible plant seed oils. There comes the food versus fuel debate, as we cannot use those oils because they will compete with the human food needs. After that comes the second generation of oil, which were the non-edible plant oils. They were also limited because the cultivation can lead to deforestation, as there are land usage, huge amounts of land usage. Next is the like currently which we are making and also in market is the bio oil, which is made from microalgae. It is the most ideal currently as it grows independent of land usage or resources, and it avoids the drawbacks of the previous generation oils. This is our first problem which we are addressing. The second problem is the methods of cultivation which were traditionally present. The methods of cultivation were open pond system, tubular photobioreactor, and tabletop photobioreactor. The open pond systems basically are where the microalgae are grown in open ponds. The open pond systems suffer from contamination as there is no security whatsoever. Coming to the tubular photobioreactor, here there are huge and thick amounts of big tubules which are used for the cultivation. We also cannot implement it on a longer basis or on a like in the market we cannot introduce it because it is very expensive to install and maintain also. Coming to the tabletop photobioreactors which are currently existing, they only produce the algae biomass on a very small scale. So the growing need for renewable carbon neutral resources highlights microalgae as a very promising solution. As I have mentioned about the problems and cons of the other cultivating methods, our one is a tabletop photobioreactor, but it is a automated and portable, which is a novelty as well. And it will yield a maximum amount of biomass with high lipid content. As I have mentioned, lipid only comes because we are cultivating our microalgae because of producing bio. So this is a comparison table of all the dielectric properties of different insulating oils. And as we can see from all the properties like dielectric strength, relative permittivity, dissipation factor, and everything, these are all the properties which we will see we will see in any oil when it is used for insulating purposes. So as we can see, the algae bio oil often exceeds all the other oils. And of course, it is very sustainable. That's why we are shifting to this bio oil. So already mentioned, our problems were open ponds and basic reactors. They lack stability and automation. The existing photobioreactors are very costly and complex. Monitoring the algae growth also becomes a problem as algae requires many factors and many components for its growth. Current extraction and biofuel methods are efficient but very expensive. So our main objectives are two objectives, which are to design and develop a novel portable tabletop algae cultivator and to harvest the biomass that will be formed from the bioreactor and process the biomass to make bio oil, which is suitable for dielectric materials and transformer insulating purposes. So how we are cultivating this bio oil? This is a methodology that I have provided. On the left side, as you can see, is a pictorial description, and on the right side is the same thing explained in words. So basically, as you can see, this is a bioreactor vessel in which algae, along with Pg11 medium, will be introduced in the bioreactor. The sensors and actuators are present on the top and bottom, respectively. The sensors which we are using are temperature sensor, carbon sensor, lux sensor, and pH sensor. These are the essential things which algae needs. All these components like temperature, pH should be in their optimum levels only, and only then can the algae grow optimally. Next comes the RGB camera, which is also deployed on top of the bioreactor. The RGB camera will acquire pictures from time to time of the algae which is growing inside, because algae, as we know, of course, it grows by photosynthesis. It makes its food. So algae has a generally greenish color. So if the color changes, we can observe it through the camera, and we can instruct the actuator, like not we, the microcontroller which is present can instruct the actuators to function, and the nutrient bottles will open and the respective nutrient which is required will be pumped in. This is a fully closed loop threshold based system, and there is no human intervention whatsoever. So this is my methodology which we have adopted. Next, this is basically what we are doing. Next comes our results and discussions. This is the bioreactor which we have currently employed in our lab. And this is where all the sensors, as you can see, this is the pH sensor, and all the other sensors are employed on the top. And this is the algae and BG11 media. So this is where the microcontroller which we are using is ESP32. And this is all my connections and all the sensors connections which I have done. And these are all the sensors readings which we have acquired by the DAC module, which is Analog Discovery 2 module, which we have used for acquiring all the information and all the sensor readings. Next comes, as I have mentioned, that we are going to make bio oil. So basically what happens after we have grown this algae for over a period of 8 months, we will deploy a filtration unit beside the bioreactor. That filtration unit will filter the biomass which is present, which will be produced in the bioreactor. That biomass which will be filtered will be taken out, and then we will dry it. How we dry it is by two methods, either by sun drying or oven drying. Oven drying is a little bit complex, but we can also use that. It is no problem. After drying, we will take, like for our application, we have taken 2 grams of the dried algae biomass, and then to it, as you can see in the left-hand side picture, to it we have added a solvent mixture. The solvent mixture is basically 2:1 ratio of chloroform and methanol. Basically, 300 ml of chloroform and 150 ml of methanol is added as solvent to the dried biomass, and it is heated in the heating mantle for 2 hours. After that, the distillation process takes place in the heating mantle. Then the extraction process, basically it separates the oil and the other solvent, and then the solvent is evaporated, and as you can see on the right hand side, the crude bio oil is produced. We have tested it for various dielectric properties such as the breakdown voltage and viscosity and thermal stability, and it often, like we have noticed, it is much better than the currently present oils. These are all my references I have used, and thank you for your cooperation.